Well, welcome everyone to the latest, the last for this calendar year in the series of NAI Director Seminars, and we are very fortunate to have Professor Roger Summons from MIT speaking to us today. And just to demonstrate the inter-team collaboration, he is speaking from JPL, where, of course, we have two new teams that have just come on board, or just coming on board, the Institute. Roger is the head of the geobiology group at MIT. He's a very distinguished scientist who I think probably needs almost no introduction to this group. I will mention that he's a fellow of the Royal Society. Comes to us originally from the Australian National University and the University in New South Wales. And he is going to be speaking to us this morning about what has been called the mother of all extinctions, the Permian-Triassic extinction, about 250 million years ago. And his title is The Great Mass Extinction, a Sudden Event or a Slow-Moving Train Wreck. And Roger, without further ado, I'll turn it over to you.
Thank you very much, Carl. Perhaps I should explain the title a little bit better. As I'm sure many of you are aware, this is the largest mass extinction that's recorded in the geological record, and a lot of the hypotheses about this extinction point or suggest that it is a really sudden event. I was explaining my data to a colleague, John Hayes, and he said, I think a better description is slow-moving train wreck. So that's the origin of the title. The work I'm going to speak about involves many people. In particular, a group from the Nanjing Institute of Geology and Paleontology in Nanjing, China. Myself, Gordon Love, and Lindsay Hayes, working from MIT, and then a number of colleagues from around the world who've provided geological sections for this event in different sites. I should also say that the work was funded almost entirely by the NASA exobiology program, and I'm very grateful for the support of the exobiology program over many years. I will start by giving a little outline on biomarkers and proxies for biogeochemical processes. In particular, I'm going to talk about biomarkers for the green sulfur bacteria, coupling of the carbon and sulfur cycles, and then I'll talk about this event as it's recorded at different places around the world, and particularly the event as recorded at the type section in South China. We've published one paper on this particular event. The first author is Clea Grice from Curtin University. That appeared in Science in 2005. Basically, this is an update of that paper. First of all, the biomarker principle. The idea embodied here is that we can use lipids as proxies for organisms because many organisms make diagnostic lipids. The importance of focusing on lipids is that they are geologically stable, whereas amino acids and DNA are easily destroyed very soon after an organism dies. Lipids are quite recalcitrant. They undergo a process known as diagenesis, where they lose certain functional groups, but the core lipid, or the core skeleton of the lipid, for example, here's dianosterol, a lipid that's found in dinoflagellates. The hydrocarbon skeleton preserved in the geological record is a hydrocarbon dinosterane. The other aspect about this is that we can find dinosterol in modern dinoflagellates. We can find a marked increase in the concentration of dinosterol in sediments in the middle Triassic, when there's the major radiation of dinoflagellates. But we can find dinosterol or dinosterane in sediments that are in fact quite old, and perhaps we can make a leap of, faith or judgment here, and suggest that this hydrocarbon might be coming from the ancestors of modern dinoflagellates, and particularly some of the strange ornamented acritarchs that we find in the Neoproterozoic rock record. The other idea embodied in this picture is that a sterol, when it's made by a microbe or an alga, has a very defined stereochemistry. That is, if you make a plane through the steroid ring system, the methyl groups and the alcohol substituent are either up or down with respect to the plane of the molecule itself. And there's a single stereochemistry in a biolipid. But when that molecule becomes buried and undergoes thermal alteration, there are numerous positions in the molecule that undergo rearrangement. And they do it in a very specific way. So you can use a stereochemistry of the product to give you some information about the thermal history of the sediment. And you can also use this geological stereochemistry to distinguish between modern lipids and ancient counterparts, because you can only get the ancient counterpart through a long period of time of heating when buried in rocks. The analytical methodology we use, basically we're looking for fresh, clean, and particularly organic rich rocks. We extract those rocks with solvents, extract out the lipids. We get a bitumen, which is a very complex mixture. We clean that up by different kinds of chromatography, particularly liquid chromatography, and we separate out different fractions of hydrocarbons based on their polarity. And then we analyse those products using a high sensitivity, high resolution GCMS system that gives us both identifications as well as quantitative information, provided we've got the right kinds of internal standards. And some examples of lipids that we use in particular, and some that I'll talk about a bit later on. Bacteria hopanes are a very important class of phospholipids, come from the bacteria hopane polyols of different kinds of bacteria. Sometimes they have distinguishing features, such as this compound here as an extra methyl group at this position here, position 3. So these are 3-methyl hopanes. Predominant source is probably bacteria that eat methane for a living. If we have a methyl group, on the other hand, at position 2 here, this lipid is found in many kinds of cyanobacteria. Roughly 65% of the cyanobacteria, 35% of the cyanobacteria we studied make this particular lipid. And then algae in particular make some distinctive compounds, and because there have been major radiations and changes in algal communities over geological time, sometimes these particular molecules are very useful as age diagnostic biomarkers, because they appear in the fossil record, the molecules appear in the fossil record at about the same time as we see microfossils representing some major groups. Carotenoids are also very useful compounds, particularly in modern organisms. Many photosynthetic organisms, in fact, make distinctive carotenoids. Things like beta-carotene or lycopene, which are shown on this slide, are quite non-specific because, for example, cyanobacteria and higher plants will both make beta-carotene. On the other hand, carotenoids which have aromatic ring systems are in fact restricted in their distributions. And I show here 2 carotenoids that are found in the phototrophic green sulfur bacteria. And note that they have a particular arrangement of the methyl groups around the ring. This one is 2, 3, and 6. The second group of aromatic carotenoids have a different arrangement of methyl groups, 2, 3, 4, and they're found in the purple sulfur bacteria. They're also found in sponges which appear to have purple sulfur bacteria as symbionts. So these 2 groups of carotenoids are largely confined to 2 very specific kinds of sulfur bacteria. With respect to coupling of the carbon and sulfur cycles, this is just a cartoon showing modern ocean and processes go on in the modern ocean. The main one that's important to us, of course, being oxygenic photosynthesis. CO2 and water give biomass and molecular oxygen. What we observe in the modern ocean is that roughly 99% of that organic matter is rapidly recycled through the process of respiration. So other microbes, plankton, fish, etc., eat that organic matter and respire it, and the cycle turns. But a small amount of the organic matter will actually sink and escape into the sediments, and a small amount of the oxygen that's produced in photosynthesis will escape to the atmosphere. Once in the sediments, organic matter is respired by the process of sulfate reduction, which is shown by this equation. And basically, probably 99.9% of organic matter disappears in these two kinds of respiration. The small amount is preserved in sediments. If you actually increase the amount of organic matter that's sinking, then sulfate reduction can actually move out of the sediments. As oxygen is depleted in the deeper waters, the zone of sulfate reduction can move up into the water column. In fact, a classic example of water column like this would be the Cariaco Basin. Other places would be basins off the California borderland, Ventura Basin, for example. Now, if you have a really large amount of organic matter into the sediments, you can, in fact, bring the zone of sulfate reduction up into the photic zone. And that's shown here. We call this a euxinic water column, and a classic example in the modern world would, of course, be the modern-day Black Sea. And the euxinic water column is in fact stabilised because the lower layer is saline water that's come through the Bosphorus, and the surface layer is largely fresh water that's coming from the Danube. So there's a very stable euxinic water column in this case. Now, the green sulfur bacteria have very distinctive physiology. Basically, they carry out anoxygenic photosynthesis, shown here. They have absolute requirement for light and hydrogen sulfide, which is the electron donor for photosynthesis. So the hydrogen sulfide replaces the water that aerobic organisms are using, oxygenic photosynthesis is using. Now, there are two kinds of green sulfur bacteria. There's what's called a brown pigmented form, which can use low light, relatively low light, and a green pigmented form, which requires higher light intensities, and the light harvesting of these two systems is controlled by their pigment compositions. And the green sulfur bacteria, as I said before, produce these carotenoids, isomania retina and chlorobactene. And these compounds, in fact, have hydrocarbon, stable hydrocarbon products, isomania and chlorobactene. And in fact, the preservation of these pigments is enhanced by the environment in which the pigments are formed. In a sulfidic water column, this highly unsaturated centre of the carotenoid ring system becomes sulfurized. It reacts with the hydrogen sulfide that's also in the water column. It reduces the double bonds, and it produces a very highly stable carotenoid pigment. It's not a pigment anymore. It's preserving the aromatic ring system, but the chain along the middle is in fact saturated. And then we can identify this particular compound and its other diagenetic products, the fragments of this molecule, by focusing on just two ions in the mass spectrum, the diagnostic ions that come from this particular fragment here, ions with mass 133 or 134. So it's a very nice system to illustrate use of biomarkers because actually everything goes together. The formation of the molecules as well as the preservation system that allows us to find fossils in rocks as old as 1.7 billion years. Now turn to the mass extinction itself. These are fairly old depictions of the mass extinction that were published by Jack Sepkoski and two papers in, 1996. And the diagram shows geological time starting from the Precambrian here, and the number of families of marine invertebrates. And we can see the major radiation of invertebrates during the Cambrian. We can see the major radiation in the Ordovician. In the Ordovician, we can see a drop in diversity here. We can see another drop in diversity here in the Devonian, where there's a significant mass extinction. We go along here is the Permian-Triassic mass extinction, which, as you see, looks like a very sudden event, and it's the largest loss of biodiversity. There's a slow recovery. There's another mass extinction at the end of the Triassic. Again, increasing diversity up through the Mesozoic until the end of the Cretaceous, and of course we have the other very well-known mass extinction, the one at the end of the Cretaceous. As I said early on, there are a great variety of hypotheses about what caused this event. They fall into two classes. The catastrophic type, such as ideas that came in the 1970s that was caused by the Earth coming in contact with radiation from a supernova. As recently as 2001, Luann Becker's hypothesised that the agent of extinction is a bolide impact, similar to what happened at the end of the Cretaceous. The other kind of catastrophic cause is major volcanism. The Siberian traps erupted massively at about this time, producing large amounts of carbon dioxide and perhaps other toxic gases into the atmosphere. And numerous people have hypothesised that this is in fact the cause. But there are causes in addition that people have hypothesised based on geological record. For example, climate change, lowering of sea level. Many people have recognised signals for ocean stagnation and marine anoxia. Excuse me. There's a major negative carbon isotope excursion that marks extinction, and people hypothesise this is caused by catastrophic collapse of methane hydrates in the ocean floor, and that has been in fact what's driven dramatic climate change. And the other hypothesis is that we have an overturn of the euxinic ocean. In fact, the toxic agents are CO2 and hydrogen sulfide released by that. And as Doug Irwin has often said, well, it's not just one cause, it's in fact a combination of these things, the so-called tangled web of causality. So, but what about the boundary itself? Well, there's a severe mass extinction at the end of the Permian worldwide. It's the end of the Changxingian stage. The boundary itself is not defined by the extinction, but it's defined by a fossil that marks the beginning of the Triassic period, or the beginning of the Mesozoic. And that's a conodont called Hindiotus parvus, and the type section that's used worldwide for people to correlate to this event is in Maishan and China. There's also an extinction on land. There's a well-known coal gap. There's a lot of coal produced through the Carboniferous and the Permian, and all of a sudden you reach beds where you have red beds and absolutely no coal. In Australia, it's defined as the end of the coal, as well as a particular kind of plant Glossopteris that's widely distributed through the later part of the Permian. Now, one of the problems, or one of the issues that's created a lot of difficulty in understanding this extinction is there's no commonly agreed way to correlate the marine and the terrestrial extinction. There are very poor dates for the loss of terrestrial organisms, and there's no fossil that occurs both in marine and terrestrial environments. So this caused a lot of argument, in fact, about where the boundary exists on land, and it's a very important problem that really needs to be addressed. There's also a faunal extinction in the terrestrial environment that's been described in particularly by Peter Ward. So this is a complicated diagram, and it basically shows the work that's been conducted by Professor Jin-Yu Gan, or the late Professor Jin-Yu Gan and his group working in Nanjing. And basically it shows here the demise of major taxonomic groups, and that occurs in what's called bed 25 and 26 of the Maishan section. But the red line, in fact, shows what's formally defined as the Triassic, the beginning of the Triassic. So this is a little bit later than the actual mass extinction itself, or the main phase of mass extinction. And the main extinction horizon, I said, is beds 25 and 26. And that age of, or that particular event is also marked by an ash bed at Maishan, which is one of the reasons Maishan was selected as a type section, because the volcanic ash bed with zircons that have been dated very accurately now by my colleague Sam Bowring and also by others. So there's a now pretty common agreement that this event took place 252.2 million years ago. The type section itself is now a geopark in Maishan. So this is a monument to the mass extinction. Here's a ginormous version of the conodont, which is basically the tooth part of an enigmatic fish of some kind, perhaps. There's some geotourists, and here is Professor Jin-Yu Gan, who's basically led the definition of the event at Maishan. And the actual type section is up on this rock face here, so you can actually walk along a path here through the last part of the Permian till you come to the very end of the Permian. There's actually a golden spike that's stuck into into the rock face there, and that's the official boundary. Now, since it's a geopark and it's a defined geological site or site of great geological interest, you can't take samples from here anymore. Very early on, you could actually collect from here, but now it's impossible to do that. So you have to collect from sections of this that are several kilometres away. And in 2004, Professor Jin organised a drilling project, and this was because the actual outcrop is in fact quite heavily weathered, and in order to get fresh samples to do a really good geochemical study, Professor Jin organised a drilling of, in fact, several cores, and our samples that I'll talk about now come from what's called the Maishan 1 core. And the core was actually drilled and samples collected by my very good colleague Chao Chen Chun. We looked at roughly 120 samples from the late Permian through the mid-Triassic, and we measured bulk geochemical parameters as well as detailed lipid biomarkers. So I'm going to show you some plots of the data we've got. So here is, now it's probably too small to see, but basically this is the Maishan stratigraphy. The extinction horizon is marked by the little yellow zone here, and then we have the beginning of the Triassic at this point here. There's quite distinct changes in lithology as you go up through the sedimentary package. The sediments in the late Permian are largely carbonates, and they're replaced very suddenly at the boundary by clastic sediments. And this is the relative sea level curve that's been drawn for Maishan. In fact, right at the extinction event is a sea level low, and that is also recognised the world over. So the next plot is showing the distribution of strontium isotopes. Without going into great detail, this increase in the strontium isotope ratio just here is a signal for weathering. In fact, what it represents is strontium from granites entering into the ocean, and that's a signal that basically there's a weathering event associated with this. And these ages here are all from individual aspects that occur through the late Permian, showing that we now have very, very good time control of the events leading up into the event itself. There's about 1.5 million years embodied in this last part of the Permian, of Chengxinyan, and then there's another 1 million or 1.5 million embodied in the section up through the first part of the Triassic. So there's roughly 3 million years embodied in the sediments that I'll be talking about. As I said, the event is marked by a major carbon isotopic excursion, and that is shown just here. In fact, correlates very nicely with this strontium isotope curve. And overlain on the slower carbon isotope excursion, which is the thing most people measure, is a very, very sharp excursion here. So this is in fact a double excursion. There's events going on 2 timescales shown by this particular curve. So that's the carbonate carbon. Here's the organic carbon. There's again a very, very sharp spike. Another interesting aspect of this is that the sharp spike in organic carbon is not exactly at the same time as a sharp spike in carbonate carbon. So again, there's an anomaly in the timescale of the residence times of organic carbon and carbonate carbon in the ocean. That may be useful later in understanding the mechanisms of these isotopic changes. And next is the nitrogen isotope curve. And the important thing to observe here is that we are going from the late Permian with values that are around about +2 to +3. And that is very typical of what the values of nitrate that you find in the ocean. So in the Permian, we have values that are typical of what you find in the modern ocean, but as we get close to the boundary, you can see there's a very gradual trend towards lower values, and right at the boundary is a sharp spike, and at that point the del 15N of sedimentary organic matter goes negative. And that tells you that there's been a major change in the nitrogen cycle, and particularly that we've likely snuffed out nitrogen cycling through nitrate, and that probably the source of organic nitrogen at these points when it's 0 to negative is in fact nitrogen fixation. We've identified the presence of the distinctive carotenoid isorenia attain at the PTB. So this is a chromatogram of the diagnostic iron 1, 3, 4, and it shows here distribution of what we call aryl isoprenoids, so they're fragments of this larger molecule. And up here we have the tiny little peak you can see, which is in fact the intact C40 compound isorenioretane, and we know that for sure because we've run standards, so we've compared the sample with the little peak with a mixture of reference carotenoids, and we're absolutely certain that identification is correct. And of course this is an unambiguous marker for the brown pigmented version of the green sulfur bacteria, and that shows that hydrogen sulfide must have come within 80 to 100 meters of the sediment surface, because that's a zone where the green sulfur bacteria live in the modern day Black Sea and other places. They basically operate quite well at low light intensities, and that's the zone where we generally find them. So now we have some biomarker records at Maishan, and the first one is the aryl isoprenoids. And what you can see is they actually first appear in the record right down here in the Lung Tan formation. Then gradually the relative abundance of these compounds, and this is absolute concentration, part per million of total organic carbon, and you can see a major spike in the presence of aryl isoprenoids here in the middle of the Changxingian. We get some very, very large spikes around the boundary, and in fact you can still find these compounds in sediments right up into the early part of the Triassic. The actual intact carotenoid follows this somewhat closely. So here we have the major peak of isorenioretane, again ppm of TOC, and again some sharp spikes at the boundary. So we have a record of marine euxinia that predates the mass extinction by at least a million and a half years. This is a Homo hopane index. Without going into the detail of how we derive it, basically it's a marker for transport of organic matter under euxinic conditions, and the higher the value, the more euxinic the water column, and we can see the highest Homo hopane index again through the Changxingian stage. In fact, the whole of the Changxingian looks like it's a euxinic, it has a euxinic water column here at Maishan. And this is the pristine phytane ratio. Again, that's another marker for anoxicity. The lower, again, we have very low values. That's more indicative of more reducing conditions through the Permian. And in fact, once we get into the Triassic, we have some very significant spikes. So there's a lot of changes in the redox state of the water column going on in the basal part of the Triassic. Here's some biomarkers for microbial physiology. The curve shown here is the 2-methylhopane index, and I said before that's a proxy for the abundance of cyanobacteria. It goes along at a fairly normal value. I think it's about 8 or so % of all hopanoids up till we get to the boundary, and then we see some very, very large values of the 2-methylhopane index, suggesting that at and after the boundary we in fact have proliferation of cyanobacteria. This is a 3-methylhopane index. In fact, these values are also quite high compared to other parts of the Phanerozoic geological record, suggesting there is an active methane cycle going on in the water column at Maishan. This one is proportions of steroids. Now, the changes in this particular parameter represent changes in the algal population. But I think the most significant result is shown in this last curve. This is the hopane to sterane ratio, and it's a rough proxy for the proportion of phototrophs in the water column that are bacteria versus algae. Okay, and you can see there are two major spikes in the hopane to sterane index. There's this one during the Changxingian, and there's this other one that basically takes up the first part of the Triassic. So what I interpret from this is that instead of having an algal ocean, as we largely have today, well, of course we have abundant cyanobacteria in the deep ocean, but primary producers in the shallow ocean are largely algae. But at this particular time, we have basically a bacterial community being the major source of new photosynthetic carbon just after the extinction event. And in fact, the values of this particular parameter you don't see in the geological record until you go back into the Mesoproterozoic. So these are in fact unprecedented for any sediments in the Phanerozoic record as far as I'm aware. Maturity. All the sediments at Maishan have been roughly buried to the same depth, and we expect they should have a common maturity, and that's shown by this particular graph here, which is representing one of the hopanoid maturity parameters, and you can see it's fairly constant, so that looks good. This is the steroid maturity parameter, and you can see this fluctuates a lot, but it fluctuates mostly at the same time that we had high hopanoids. In fact, the fluctuations is not a real signal. This is, in fact, the signal is being corrupted by the high amounts of bacteriohopanes. Now we've got a couple of parameters here for other kinds of hopanoids, and these are varying in very significant ways, and this is quite enigmatic. If one particular maturity parameter shows a fairly constant value, and these other ones are showing these major fluctuations, it shows there's some very unusual input of bacterial hopanoids to the sediments. And yeah, I think I should leave it at that point, because I guess not a lot of people are interested in these arcane aspects of the biomarker distributions. But in fact, what I would say about this is some very important signals of the types of organisms that are present in the water column at this time. Now, I spoke before about the nitrogen isotopic composition of organic matter at these sediments, and here is a blow-up expansion of the del 15N curve for just around the boundary. So here we have the fairly heavy values of the late Permian. We go down to 0 here, and then we have these big fluctuations that go into negative territory, and these are the peaks of the 2-methylhopane index. It's possible that the source of newly fixed nitrogen is in fact diazotrophic cyanobacteria, but the peaks in 2-methylhopanes don't exactly correlate with the most negative values of del 15N, but what does is the peaks in aryl isoprenoids. So we get these light values of del 15N when we have the most euxinic water column conditions. The boundary, as I said, the boundary event has a number of hypothesized causes. One of the most prominently published causes or hypotheses is that it was a bolide impact. 2 papers in Science, first of all referring to the presence of noble gases with an extraterrestrial signature encased in fullerenes, the boundary, and also the identification of a possible impact crater off northwestern Australia. The case for a mass extinction caused by a bolide at this time, I think, is very tenuous. So we've been looking at this particular evidence in some detail. Interestingly, just a few weeks ago, there was a paper appeared in Geochemical identifying the source of strong fullerene envelope arising from laser desorption mass spectrometric analysis of meteoritic insoluble organic matter by Hammond and Zare. And the technique that was used in these earlier studies to find the fullerenes was laser desorption mass spectrometry. And fullerenes have been reported in several places in the geological record, but there's also been identified in meteorites and interplanetary dust particles. And Hammond and Zare, in fact, have shown that the presence of fullerene could in fact be an artifact of the mass spectrometric method which was used to detect the fullerenes in the first place. So we've done a study of Maishan sediments looking for fullerenes, and we used a different kind of mass spectrometry. We used electrospray to ionise the sample, and TOF, time of flight mass spectrometry, to analyse the products. And we obtained a standard of fullerene, and the spectrum is shown here. So this ion cluster here represents the molecule C60. There's a very distinctive mass because it's 60 times 12.000, and there's a very distinctive combination of isotopes because 1.1% of all the carbon in this molecule is carbon-13, and this particular isotope cluster follows that rule exactly. And then fullerene is prone to oxidation, so you see fullerene with one oxygen and a second oxygen. And we're able to detect fullerene using this method. We can detect as little as 3.5 picograms, so we have a very low detection limit. We also were able to obtain a sample of fullerene with slight enrichment, I think 15% or 20% carbon-13, and that's the spectrum shown here. And we actually added this to our extracts of the sediment to act as a carrier and an internal standard. And in fact, this is a sample of the C13 enriched fullerene from an extract from Maishan bed 25 at the Extinction. In fact, what you find is that you can find your carbon-13 labeled material, but you can't find any natural abundance C12. So basically, I think we've disproved the occurrence of fullerene at the boundary at Maishan. Now, in order to understand the event, there's no point just looking at Maishan, because it's a big world. You can have quite different environments along different parts of continental margins. So now we can take advantage of the wonderful paleogeographic maps. I'll show you some from Ron Blakey, Northern Arizona University, and we can rotate the continents back to the way they were at this particular event, roughly 250, 260 million years ago. So that's the arrangement of the continents at 260. Now I'm going to show you roughly 255, and this is now a Scotese map, but it's very similar. And the section at Maishan, Maishan is just here, and so you can see that it's on the quite close to the equator, and it's on the margin of this Paleotethys Ocean. Previously, we've looked at samples from the Perth Basin in Western Australia, which is down here. So this is in quite a high southerly latitude, but it's also in the Paleotethys Ocean. And again, we found abundant aryl isoprenoids. This is material from the science paper a couple of years ago. I won't go into it in great detail, but basically, as you get to the PTB, you can find marked increases in the abundance of aryl isoprenoids. There are also major isotopic anomalies with respect to pyrite. So there's a sulfur isotope signal at this event that I really haven't spoken about, but you can observe that in the sediments in Perth Basin, Western Australia. Another well-documented location of the event is in modern-day Tibet. That's actually in the central part of the Tethys Ocean. We've analysed some sediments that came from Richard Twitchett. There's chromatograms here, so the bottom one is our reference standard of aryl isoprenoids, and these are all samples from the Tibet outcrop, and we can identify the presence of aryl isoprenoids or the products of the breakdown of isorenioretane. There was a great paper several years ago by Jonathan Payne, Andy Nolan colleagues, that described the demise of organisms in a carbonate reef system at this particular event. And the biostratigraphy is shown here. This is the boundary down here. And what you can see is the carbon isotopic signal that we see at Maishan is only one of a number of major carbon isotope anomalies in the early part of the Triassic. We've looked at the Great Bank of Guizhou, which is located here again in the Tethys, and here's some more chromatograms. This time the standard's on the top, and these two chromatograms are for showing the presence of aryl isoprenoids that are actually preserved at the edge of the carbonate reef system. So several places now in the Paleotethys Ocean we have the same signal at exactly the same time. All of these places are correlated well with being at the boundary by the presence of the diagnostic marine fossil, Indiotis parvus. So what's going on the rest of the ocean? Well, here's a section from Peace River. So this is in Western Canada. On the map, it's roughly at this position here. The section, there's no complete section, geological section for the event, but with the help of colleague Charles Henderson and his student, we've been able to reconstruct a PT boundary section by piecing together pieces of different oil exploration wells, because this is a petroleum province, so we've been looking through a number of cores of oil well or petroleum exploration wells. and basically pieced together a stratigraphy, and again been able to identify the exactly same signal that we get at Maishan for these smaller aryl isoprenoids. And here are the C40 compounds up here, and in particular I point to this compound here, chlorobactane. Now that's from the diagnostic pigment of the green pigmented form of the green sulfur bacteria, and these guys absolutely require more light than the brown pigmented forms, and that shows or suggests that the water column here, that the euxinia was in fact, the euxinic zone came higher and closer to the surface, maybe as close as 30 or 40 meters of the surface. So intense a signal for intense euxinia here as well. Cap Stotch, this is a classic section. Now we're going to the boreal ocean. There's a lot of terrestrial organic matter in this particular section. It was described as long ago as 1972. 2-methylhopane biomarkers. I'll just speak very briefly about this. It's kind of controversial. There are potentially other sources of these compounds, but the very high values that we find at Maishan are part of a pattern that we see through the geological record. Now here's a picture of the 2-methylhopane index. So this is representing the proportion of 2-methylhopanes as a fraction of all the hopanoids. And here we have geological time for the Phanerozoic, or the last 600 million years. Most of this data is coming from petroleum samples that have been analysed. And what you can see is you get very high values at the late Devonian mass extinction. You get very high values at an oceanic anoxic event in the Triassian. There are several oceanic anoxic events in the Cretaceous. They have high values. The highest value of all is the one that we measured for the Permian-Triassic section at Maishan. So there's a systematic observation here that these particular biomarkers are highest when we have these major oceanic anoxic events, which are characterized by disruption of the nitrogen cycle, and perhaps open a window for cyanobacteria, or give an advantage to cyanobacteria that can fix their own nitrogen. So, stagnant oceans, they're toxic to all animals, of course. Bacteria, on the other hand, can survive. So when you have a stagnant ocean, you have low oxygen, you have high PCO2 and high amounts of H2S, and all have adverse impacts on metazoa. In Cleedy's paper of 2005, we hypothesized that one of the agents of extinction, or major agent, was the spread of anoxic and sulfidic waters onto continental shelves. And other people have made similar observation, particularly Lee Kump, who's done, and his colleagues Pavlov Arthur and Katja Meyer, who've modelled this. And in fact, if you can put sufficient hydrogen sulfide into the atmosphere, in fact it could be quite long-lived, because the mechanisms by which you can remove the hydrogen sulfide are complex and take significant time to in fact clean the atmosphere. So we have a carbon cycle operating in a stratified ocean, and we have a euxinic water column, and at certain times hydrogen sulfide evading to the atmosphere and perhaps adversely impacting the terrestrial biosphere. We in fact have some modern counterparts. This is a satellite image of water column off southern Namibia, where it's well known that you have events of sulfide eruption that cause major fish kills. And the sulfide eruption, in fact, is visualized here in the presence of these sort of green clouds, these clouds here, which are in fact elemental sulfur that's been formed from the oxidation of hydrogen sulfide. And of course, the other very well known example is the dead zone that we see in the Gulf of Mexico, caused by agricultural pollution coming down the Mississippi River, and of course the dead zone again, the demise of fish and other organisms driven by high concentrations of hydrogen sulfide. So to summarise everything, hydrogen sulfide in the ocean atmosphere would be toxic to all but bacterial life. Understanding the causes of this complex web of causality, I think, can be connected to the long time scale of this event. I think it's related to the aggregation of the supercontinent Pangaea in a greenhouse world. When you have a greenhouse world, you have a low equator to pole temperature differential, and that helps the ocean circulation to be very sluggish, in fact much more sluggish than today when we have ice at the poles. The uplift associated with the continental aggregation could lead to the massive weathering of the Paleozoic coals, and this would draw down oxygen, elevate CO2, and the organic matter transferred to the ocean could in fact provide source of food for sulfate reduction by sulfate reducing bacteria. A tectonic underpinning of the event accounts for the delay in the recovery. Euxinia waxes and wanes, and a long time is needed to re-ventilate the ocean. I think it also accounts for repeated carbon-13 excursions, but also the different pace and scale of the excursions at the different locations where it's been measured. And the biological consequences could have been made worse by the intense volcanism that occurred near to the boundary, but we can see from the Euxinia, or the signal for Euxinia, this event was presaged by a long period of a toxic ocean. And in fact, if you look closely at this particular figure here from one of Andy Knoll's papers, you can in fact see there is in fact an extinction in the end of the Middle Permian, and there's a continued loss of biodiversity. So it may be a sudden event at the boundary itself, but you can see in fact there's significant loss of biodiversity leading up to the main extinction event. And there's one more, I think. Yes, acknowledge my colleagues, particularly Doug Irwin and Professor Jinyu Gan, who had the foresight to study this event. It's so well documented now at Maishan, China, and the people who did all the hard work for sampling the core and a lot of the analytical work, Chao Chen Chun, he was assisted and in fact guided to a larger degree by my good colleague Gordon Love, and other sections have been studied by clearly Grice, of course, Western Australia. Lindsay Hayes is doing the work at MIT now, and in fact Emma Grosjean, who's now at Geoscience Australia, worked from the beginning to set up the laboratory so we could in fact do this work. And I'll thank you all for your attention.
Roger, thank you very much for a great talk. We'll open it up for questions now, so if you have a question, please raise your hand in WebEx, and I'll give you just a second to do that. This is the last director seminar of this calendar year. Normally we'd pick up director seminars again at the end of January. I think we're probably going to delay it a couple of months because we're going to have a series of video cast seminars by all the 14 principal investigators in the Institute. And I should have mentioned in introducing Roger, of course, that he is a principal investigator of our MIT team, where he's focused on an earlier period in Earth's biological history, and that is the development of complexity in the Proterozoic. So, Marco, do we have any hands raised?
Yes, we have a question at the Lunar Science Institute.
Yeah.
Hello, everybody. The Lunar Science Institute is very happy to be part of this discussion now. Roger, that was great. I wonder if I could just ask you to say a little more about the possible role of volcanism, or whatever was precipitous that ended this. What triggered the final event before the recovery started?
Well, I think at the present time, it's impossible to define the exact role of the volcanism, because the onset of volcanism, the time of the onset of volcanism, is not as precisely known as the boundary itself. So when you have a major tectonic episode like aggregation of Pangaea, of course you're going to have volcanism associated with that, but the timing, I think, is critical, and I know there are a number of people trying to date that the Siberian traps and the onset of the Siberian traps much more precisely, and I think at that point we'd be able to say more clearly what the role of that event was in the mass extinction. that's the best I can do, I'm sorry.
Thanks.
Okay, we have a question at Ames.
Hi, Roger. This is Dave here. Sort of a related mechanism, which of course Andy Knoll talked about, was this idea about CO2, high levels of CO2. And I guess related to that also might be ocean pH. And is there any handle you could conceive of how you might expect, for example, a drop of pH in the shallow waters, which obviously would be bad for shelly creatures. Do you think there's any hope to try to constrain sort of CO2/pH associated with this event?
I hope I made it clear that I think they're all part of the same package. You won't have euxinia unless you've got anoxic ocean, and then you're going to have high concentrations of CO2. You're going to have low pH. It's in fact a toxic mix, and certainly Andy has demonstrated that that particular taxa are vulnerable. Some taxa are less vulnerable, and the degree to which they are lost is in accord with the idea that PCO2 is playing an important part. But what intrigues me is the strange events in the terrestrial biosphere that suggests it's not just CO2 in the atmosphere that's having an effect on the plants, because my colleague Clinton Foster has in fact documented some very strange effects on plant pollen. It's very hard to understand how increased atmospheric CO2 could cause such a dramatic change in the plant world. So I think there's reason to think that there's something else that's toxic in the atmosphere, and hydrogen sulfide is a likely candidate. So it's certainly a toxic mix, and certainly low pH is going to heavily constrain any organism that relies on the carbonate skeleton. But something has to be causing the loss of diversity in the plant world. And since we're commonly taught that CO2 is in fact some kind of fertilizer, it would require enormous increase in CO2 in the atmosphere, I think, to so adversely affect the plant world.
Okay, we have a question at the University of Washington.
Yeah, this is Sean Goldman at the University of Washington. My question is, how can we be sure that the certain color pigments have always been associated with the sulfur metabolic pathways that they're associated with in the modern environment, and that maybe the correlation between those two things hasn't evolved as the oxidation state of the Earth's surface has evolved?
That's a question I'm often asked. All I can say in response is that the biosynthetic pathways leading to lipids are very highly constrained to particular organisms. There's no evidence that those specific pigments are found in any other taxa other than the sulfur bacteria. Aromatic carotenoids have been recently found in some cyanobacterium, but they're different compounds and they're not likely to be confused with the ones that I've shown. Also, we have a record of these pigments, a continuous record of their occurrence in sediments, not just at oceanic anoxic events, but in particular environments where we have stratified water columns and other evidence of sulfidic water columns. So they're not just found at this event, they're found through the geological record, always found alongside other evidence for sulfidic conditions. And the record goes back quite definitely. My former student Jochen Brox has shown very nicely that in the Mesoproterozoic MacArthur Basin we can find evidence for both the green and the purple sulfur bacteria. So a continuous geological record, occurrence in modern organisms and Phanerozoic sediments in associated with geological evidence for those particular physiological conditions required by the organism. I think that's very compelling evidence that these organisms have been making these same compounds for the same purpose ever since they were invented.
So in short, you often find them correlated with separate evidence for those oceanic conditions or those water column conditions?
Yes, they're in fact they occur in many kinds of oil, usually associated with salt. And salt, of course, is usually a salt associated with some kind of water column stratification. So there's oil fields in, there's oil in Australia, there's oil in Canada in particular, there's oil in the Middle East, there's oil in Florida, there's oil in the Gulf of Mexico that have these compounds, and they're usually associated with stratigraphic layers that have all the hallmarks of being an evaporitic system. And evaporitic system, of course, you first precipitate carbonate, secondly you precipitate the gypsum, you have mechanisms to keep the water column stratified, and under those conditions that's when you find the green sulfur bacteria proliferating. Is that...
Yes, thank you.
Roger, no one has their hand raised right now in WebEx, so I'll take the opportunity to ask a question. Could you say a bit more about how global these euxinic conditions were likely to be, particularly if this was associated with the aggregation of Pangaea, and of course all of your samples are of necessity from parts of Pangaea. You have the rest of the global ocean, and were there likely to be euxinic conditions throughout the rest of the global ocean, or could there have been oxygen oases, perhaps extensive ones, across the rest of the globe?
Yes, that's very perceptive, Carl. And of course, the only parts of the ocean that are preserved are the bits of the continental margins. As you know, the ocean crust is in the main subducted and lost from the geological record. So basically, we have a record of the ocean along the edges of continents. Isazaki has, in fact, done a study of some oceanic sediments that are preserved in Japan. As far as I'm aware, they could be the only ones of this event. But what he noticed was dramatic changes in lithology at this event, as recorded in these marine cherts. And again, he interprets that in terms of this being a major anoxic, what he calls, in fact, a super anoxic event. So from the one example that I know of, where we have a mid-ocean sediment preserved, the evidence it is in fact quite distinctive in its own right, and interpreted as evidence that the mid-ocean, at least represented by this location, was euxinic as well. In fact, Isazaki, who has worked on the section in Japan, unfortunately it's too cooked to do any biomarker studies, so these have been very highly metamorphosed and are not suitable for the sorts of things we do. But he is in fact a proponent of the hypothesis that this is a super anoxic event that started in the middle of the Permian, that it's not just one event reflected at the end of the Permian, but something that has a long gestation. And our evidence to date supports that idea very well, I think.
Can I ask a follow-up question?
Sure.
Go right ahead, David.
Okay, Morrison again. Just from the fact of this severe mass extinction, doesn't that tell you something about the unlikelihood of having an oxygenic ocean out there to preserve species that apparently went extinct?
Could you say that again, please?
Sure. If this event were limited to Pangaea and the Neotethys Ocean in that area, and the rest of the Great Ocean survived, wouldn't we have not seen a mass extinction of this level?
That's right. Exactly. And the evidence I showed, we've got at least 2 sites on the Panthalastic Ocean. We've tried looking further south, sediments of California, but they're in fact too cooked. So we're confined, as far as I'm aware, to looking at the northern part of the Panthalastic Ocean. But the 2 places we've looked at both show evidence of euxinia. So that's the best we can do at the moment, I think. We have evidence that the 2 major oceans, as well as the Boreal Ocean in a narrow seaway, are euxinic, and we haven't yet found any expression of this event that doesn't show expression of euxinia. So I think it's a pretty compelling case. We have 8 sites, and I don't know if you need more than 8, but 8 I find is pretty convincing. But we have to
Do we have any other questions? If you do, you can just open your mic and chime on in.
Well, you can email me. I think Mark is keen to have the room back. Yeah, go ahead.
Hi, Roger.
Yeah.
This is Marilyn Vogel at Ames.
Hi, Marilyn.
And I'm wondering about your interpretations of the aryl isoprenoids. I mean, obviously, you have a complex web of evidence of the anoxia, but isn't it, how do you interpret aryl isoprenoids in the absence of isorenirotane precursors? Because can't they come from beta-carotene? And sort of what are your rules of thumb about whether or not you could distinguish those two sources?
My rule of thumb is that aryl isoprenoids are coming from aromatic carotenoids and not from beta-carotene. That's my rule of thumb. And isotopic evidence is entirely consistent with that. And as I showed, at probably 5 of the 8, we have the presence of the intact C40 compounds. So I don't think there's any doubt about their original origin from the aromatic carotenoids. I personally don't think that idea that it comes from beta-carotene carries much weight at all.
Okay, it sounds like we should thank our speaker once again. Roger, thank you for a great talk. And please, everybody, tune in again when these talks resume next year. And as I mentioned, they're going to resume with a series of talks. Actually, we're going to do them probably 2 per week starting in early February, with all of the NAI principal investigators presenting seminars over the course of about 7 weeks. So thank you very much, and we'll see you all then. Bye-bye.